Hello everyone, in this video we are going to talk about liver alanine. Before we start, let me introduce our group members, which consists of Nur Izazi Haziqah, Nur Izzah Khairani and Nur Insyirah. Alanine is a non-essential alpha amino acid that is used in the biosynthesis of proteins. Amino acids are organic substances containing amine and carboxylic acid functional groups attached to the central carbon atom, which is methyl group side chain. Here is the role of alanine. Alanine is used to synthesize protein in the liver, where pyruvate is used to make the glucose for muscle tissue, brain, and the central nervous system in the process of gluconeogenesis. Furthermore, it will reduce body fat and boost the immune system. In addition, administrations of alanine prevent the elevations of alanine transaminase and liver injury. Next, alanine biosynthesis. Alanine is made from pyruvate by the actions of alanine aminotransferase, which converts glutamate to alpha-ketoglutarate at the same time. This reaction is reversible under catabolic conditions. After that, the glucose-alanine cycle, also known as Khairani cycle. The liver, blood, and muscle are the organs involved in this process. Alanine released from muscle into the blood and converted in the liver into pyruvate, which amino group is removed via transamination to produce urea for excretion through the kidneys. The Khairani cycle took place in skeletal muscle. Branch amino acid catabolized. BCAT enzyme is transferred from the branch amino acid into the alpha-ketoglutarate, forming a glutamate. Purine and pyrimidine breakdown in muscle lead to ammonium production, and the ammonium is incorporated into an alpha-ketoglutarate with the enzyme glutamate dehydrogenase and forming glutamate. As these processes continue, we get a build-up of glutamate in skeletal muscle. Glutamate is then donate its amino group to a pyruvate to form alanine with the enzyme alanine aminotransferase, or ALT. ALT enzyme requires pyridoxal phosphate, which is a vitamin B6 derivative. Breakdown of glucose through glycolysis pathway form pyruvate. ALT take an amino group from the glutamate added to the pyruvate to form alanine, and in the process, the glutamate is recycled back into alpha-ketoglutarate. Then, alanine travels in the blood to the liver. Once alanine is in the liver, it can undergo a reaction with alanine aminotransferase, or ALT, and produce pyruvate. Then the carbon skeleton of pyruvate can then undergo the process of gluconeogenesis to form glucose. In this case, alanine donates its amino group to alpha-ketoglutarate, and alpha-ketoglutarate becomes glutamate. Gluconeogenesis only can occur in the liver because it has the enzyme glucose 6-phosphatase, where the skeletal muscle does not have this enzyme. The glucose is released from the liver into the blood for uptake by tissues such as muscle, which use glucose for energy. Muscle cells use the glucose through glycolysis process and generate pyruvate. The formed pyruvate is again available for transamination to reform alanine, so we see that the metabolism of alanine in the liver can lead to the production of glutamate. Then glutamate is processed by glutamate dehydrogenase to release in ammonia. The glutamate formed in the liver can be deaminated to release ammonia, and the ammonia is used in the liver for urea production. ALT blood test is needed if you are having these symptoms, which are vomiting, nausea, frequent itching, fatigue and weakness, jaundice, and etc. in order to detect liver damage. Lastly, these are the additional informations about liver alanine. These are our references, and thank you for watching.